Thank you for the introduction. Good afternoon, everyone. I'm Mingfu from the Pennsylvania State University. I'm not Carl. Carl is a PhD student in my lab. He actually planned to be here, but last Thursday, 2 hours after his thesis defense, he broke his ankle, so he had to cancel the trip. So I'm here to present the work. All right, thanks to all the speakers before me about in this session. So we know we have a good picture about single-cell RNA-seq data. So single-cell RNA-seq data can be classified in two categories. One is 3' or 5' biased, which means the reads are concentrated on one end of the molecule. For example, the technology is represented by 10x chromium, and this is the mainstream data. Widely used. 3', 5' bias data are very good at gene level analysis, but they are not suitable for like isoform level analysis simply because the reads cannot cover the whole molecule and therefore cannot be used to distinguish different isoforms expressed from the same gene. The other category is full-length coverage data. For example, they can be generated from technologies like SmartSeq2, SmartSeq3, and more recently SmartSeq3 Express. So they are not mainstream, but they are becoming more popular. And this kind of data is able to do isoform level analysis because the reads can cover the entire molecule. So we are more interested in the second one, using, for example, SmartSeq3 data to do isoform level analysis at the single cell resolution. So the particular task we are interested is transcript assembly. Specifically, the SmartSeq3 protocol can produce sequencing reads for individual cells, and then we want to develop assembly algorithm that can reconstruct the full-length expressed transcripts for individual cells. Transcript assembly is a problem that have been well studied, and there are many methods developed. For example, string-tie series, scale-up series. The list should be much longer, but I just named two as representatives. So those methods primarily designed for bulk data, but they can be used to assemble, for example, SmartSeq3 data. But they are optimized. They are not optimized. So our goal of this project is to design a tailored assembler specifically for SmartSeq3 data. Hopefully can achieve like improved accuracy comparing with the general assemblers. So let's look into the SmartSeq3 sequencing protocol. So this is a figure from that SmartSeq3 paper. So this is like mRNA. And the protocol starts with creating lots of TSOs, template switching oligos, consists of 3 barcodes, like TN5, which will be used later for segmentation. The tag is like 11-bit pair primer, and then UMI. So the TSO will be attached to one end of the molecule, and then through the reverse transcription and template switching, we get the cDNA. cDNA will be then amplified using PCR, and then the protocol will do the segmentation, which basically creates random cuts on the cDNAs, which breaks the cDNA molecules into fragments. Let's say this one, that one, and that one, and so on. So then the two ends of the fragments will be sequenced, obtaining the paired end reads. So you can see we will have 2 types of reads. One is the reads from the first fragment of the cDNA, which contains the barcodes or UMI. We call them UMI reads. Others will be called like internal reads. So this is another figure from the SmartSeq3 paper, compares the coverage of these 2 types of reads. So this blue curve shows the coverage along the molecule for the internal reads, and you can see they cover the entire molecule quite evenly. So the green curve shows the coverage along the molecule for the UMI reads, and not a surprise, they are concentrated on the 5' side of the molecule. So given RNA-seq data, a basic question is to ask whether they are strand-specific or unstranded. So are the SMART-3 data strand-specific or unstranded? So strand-specific means the reads are from the same strand of the cDNA, and there are two cases, two possibilities. One is FR, which means the first end is forward, the second end is reverse, or like RF. The first read is reverse, and the second read is forward. Unstranded means like reads can come from both strands of the cDNA. Stranded actually can be easily tested by sampling a bunch of reads from the data, and we did that. So this shows one statistics for one read, and you can see like the FR rate is 50%, RF rate is like 42%. So it seems like it's not, or like it's unstranded, because if it's like stranded specific, for example, FR, then we expect the FR rate is like 98%. But here is like about 50-50. So it seems that the data is unstranded. But we look more carefully, like looked many more cells. We always observe the FR rate is higher than the RF rate. So FR rate is about 50%, but the RF rate is about 40%. So this is not expected. If the data is really unstranded, we expect on some cells like FR is higher, and on other cells like RF is higher. But this is not the case we observed. So this is interesting. As I expected, we explored that and resolved this mystery. So the answer is UMI reads are strand specific, but internal reads are not. So they are unstranded. So we keep explore the difference of the two types of reads. So this shows the fraction of each type for individual cells. So on the data set with 192 cells, so the x-axis indexes the cells. So the y-axis shows the fraction of each type of reads. So in general, you can see there are more internal reads in cells. On average, the percentage of internal reads is 73%. So we then compare their locations. We align reads to different locations like exons, introns, intergenic, or unassigned. And we observe a big difference for the reads that are aligned to intron regions. So 19% of the internal reads are intronic, but only 11% of the UMI reads are intronic. All right, so here is the short summary about the difference between the two types of reads from the SMART-6-3 protocols. So UMI reads are strand-specific, internal reads are not. UMI reads are 5' biased, but internal reads can cover quite evenly across the molecule. UMI reads are less abundant, internal reads are more abundant. UMI reads are more like 10x style, but internal reads are more like bike-like. So therefore, we think those two types of reads should not be treated equally. They should be handled differently. So with this idea in mind, we extend GLBIO by adding a bunch of very simple heuristics, and we name the resulting tool as MRAF. So here are the heuristics we added by acknowledging that the two types of reads are different. So the first one is strand strand correction. So internal reads are not strand specific. So then we assign it a strand by borrowing the strand of the nearby UMI reads, because UMI reads are strand specific. Second, we think UMI reads are more reliable, and therefore if a gene loci contains 0 UMI reads or a tiny fraction of UMI reads, we think they probably are contamination, and therefore we will discard that gene loci. Third one, so because UMI reads are 5' biased, they are concentrated on the 5' side of the molecule. So therefore, we think it's safe to assume that the first exon, the exon at the 5' side, should contain some UMI reads or supported by some UMI reads, and we enforce that. And this is the last heuristic. which handles those, we call them intronic exons, like this one, the orange one, this green one, this red one. They are intronic exons. They are more likely to be erroneous due to the contaminated internal reads. So we designed this heuristic, very simple. So if each kind of this intronic exon is not supported by any UMI read, and this corresponding junction, like this one, has higher coverage than this intronic exon, then we simply remove it. So that's all the heuristics, and you can see they are quite ad hoc and simple. And we tested on some preliminary results. So we compare 3 methods. One is this current one, Amaranth, and we compare with 2 other assemblers, GLBIO and Strength-2, on one SmartSeq3 dataset with 192 human cells. So the x-axis shows the number of correct transcript, which is proportional to sensitivity, and the y-axis shows the precision. Each point corresponds to a cell, and those orange points corresponds to our current method, Amaranth, and you can see the points are on top of other methods, showing that Amaranth really improves precision quite substantially. We also draw the precision recall curve by ranking transcripts by their coverage. And we show the precision recall curves of the 3 methods on 4 randomly picked cells. And you can see the curve for Amaranth is above the curve for the other 2 methods, proving that its accuracy is higher. All right, to summarize, we observed that SmartSeq3 data, there are a big difference between UMI reads and internal reads, and they should be handled differently. And we did that by adding very simple heuristics, and it seems to work on some preliminary results. And this is in progress, ongoing work. So I would like to thank Carl, who has been leading this project. I hope his ankle recover soon, and also the funding agencies, NSF and NIH. Thank you for your attention. Happy to answer any questions.
Beautiful work. So I have a question. How do you identify the UMI read? Is it through the conserved TSO sequence, or look at the trimmed alignment from star or other aligner?
Yeah, from the UMI barcode. Yeah, so right, reads with that barcode will be UMI reads, and so there's a tool like the UMI can extract those reads.
I see. Thank you.